Okay, well, welcome everybody. Expected guests and surprise guests. You're in for a treat today. I'm happy to introduce our speaker today. So we're here as part of the seminar series for the Institute of Neuroscience and Neurotechnology, which is directed by Dr. McIntosh, but Randy is also a professor in BPK. So
it counts in that context.
Yeah, it worked out perfectly. Okay, so and Randy is also the BC Leadership Chair in Neuroscience and Technology Translation across the lifespan. And he's going to talk to us today about some of his work and this amazing virtual brain platform that he and many collaborators have developed. Feel free, if you're here for the seminar or the class, we're combining the two. You're in the right place.
You get course credit, Max.
Yeah. Okay, so for me, it's hard to believe that Randy and the INN Institute have only been here at SFU for 2 years. They've done amazing things already in uniting the really broad neuroscience community at SFU, and also deepening our roots in the international research community that is allowing us to further amplify our potential for really important interdisciplinary research in neuroscience here at SFU. Just want to share your screen.
I'm not logged into the. I'm not on Zoom at the moment. So let me just continue to talk. Actually, while I'm logging into Zoom, which I forgot to do, of course, I'm going to talk to you just to give you a bit of a preamble.
Awesome.
So the platform I'm going to talk about was developed about 20 years ago, actually, originally. It was developed based on I'm being very Canadian, developed based on a pub conversation, actually. And me and the major person who was the other criminal in this enterprise, Victor Yersa, he and I were talking about some of the problems that happened in neuroscience. And one of the challenges about 20 years ago was that there was a lot of efforts in pulling the brain apart in the sense of new technologies that had been developed for measuring the brain function. So there's functional MRI, which came on board then, which allowed you to measure human brain while they were doing tasks. There was new developments in structural MRI that allowed you to look at pieces of the brain, looking at white matter changes. The challenge was you could get a lot of measurements, but it was really, really difficult to put it back together. Because obviously the way your brain functions is all these things interact with one another. And so he and I were talking over a few beers and we're sketching some stuff out. We said, you know what we should do? We should build the virtual brain. And it was like, what a cool idea. So we sketched it out on a couple of napkins. And then shortly thereafter, we had a white paper, not cocktail napkins, and then developed the platform that we'll talk about a bit today. So that is the virtual brain platform.
You're online, okay?
I'm online, okay. Am I online, okay?
Yeah. Oh, yeah. Okay, well, you figure that out. I was just to add to that,
Go ahead.
Mention that so the virtual brain is a really dynamic platform, and there's a lot of potential for it to help to integrate all of the different types of neuroscience research that we do at SFU.
Mm-hmm.
So just as one example that I'm really excited about, and when my lab we work with human pluripotent stem cells that we get from patients who have genetic diseases. We study their disease in a dish, but we think about the brain really at the level of cells, and little groups of cells and tissues that we can make in a dish. So it's been really cool to see that there's actually a place for my lab and what we do in that kind of cellular-based research in the virtual brain also. So Randy and I are working on developing a project to integrate those types of cell models into the virtual brain, which he'll tell you about today is at a very different level in terms of patient imaging and what their brain looks like on a larger scale. So with that, we hope that we can, understand patients with these, genetic diseases that we really know nothing about, from the level of cells all the way up to networks in their brain, and to try to use that to develop better treatments for these patients. So I think that's just one example of a lot of the different types of research we do at SFU that can integrate really nicely with this tool platform that you're going to hear about. Oh, you're still on.
Am I still on presenter? I just
It's
I
weird.
just
It's
changed
not.
it.
It's
The Zoom is good now. It's just the.
so weird.
I don't have any embarrassing notes in this, so.
Yeah.
I'm gonna try one more thing and I'm talking. So I want to talk about my outline while I'm waiting to get this figured out. The
You go to like advanced.
ooh, where's that?
In Zoom itself.
Oh, in Zoom itself.
I don't know if it's a glitch or this will help you. Sharing. Advanced, do you do that usually? Portion of screen. And then go here.
You-
Full screen,
Okay,
and then you put the once more, one more time. And then go to Zoom.
Mhm.
You can minimize
Still looking at the wrong thing, aren't we?
that. So if you go to Zoom, you can put that box around what you want to show.
In the meantime, for those of you who are joining the we have a surprise film sent out for documenting that. And we'll start our class at, we'll take a 15-minute break.
Does anyone
We'll
know?
our class.
Do you want those?
No,
That might work too. Just do that.
So Kelly says to swap the displays first,
so what? That's what I'm
the
doing.
displays.
She'll know.
It's funny because I just did this on Monday. It's working just fine. I could pantomime the whole thing.
New ways to know.
Yeah, okay, cool. These are IT guys.
So while the IT dudes are figuring this out, let me continue the monologue. Look over here. Don't look at the screen. So as I mentioned, the platform was built about 20 years ago. A major difference in terms of how this particular software package was developed compared to a lot of other software packages that are used in sort of the open science domain that are based on on research, is that we started fairly early on in trying, pay attention, over here, don't look up there. Early on with partnering with the industry partners to help develop the software package. So from the outset, the development of the virtual brain was actually a collaboration between a large scientific group and a software company in Frankfurt. It wasn't as if we did this with the idea of actually making money from this, because that wasn't the purpose. The purpose was to actually share the software package. The challenge, of course, with research-based software packages, they usually suck in terms of things like documentation, in terms of version control, in terms of being able to just use it. So what often happens for those of us who've written software for research purposes, you write it, you don't comment anything. You just put together the code, you run your script, and you're done. And the next student comes along or a prop comes along, tries to use the package. Of course, they go, I don't know how to do this. And there's no commenting. So we intended to share this package very early on. And in doing so, we worked with a company to make sure that we had good version control, make sure we had good usability, and make sure that things were documented so that we could share the platform, we could have people contribute code and be able to contribute to the code. So what ended up happening from the outset is that the Virtual Brain platform was in fact a community project that allowed people not just within our small group to contribute software, but anybody from any place could contribute the software.
It works.
We're good?
There's one last step. We're not fully there.
Oh, okay.
Oh, the Zoom is not correct yet.
So The other unique thing about the platform was that it's an intersection between computational modeling per se and data analysis. This is a great distraction, isn't it? Because you're talking.
Yeah, sorry.
There's a dog over there. Whoo!
Yeah, my apologies.
We just need a squirrel now. You just complete the whole thing. So the platform is since I was talking about the motivation came from a discussion I was having about putting the brain back together. The question then becomes, so how do we do that? Are we ready to go? Okay. We can then take empirical data that are measured with these scanners I mentioned, with MRI, both brain structure and function, and use the computational platform to knit them together so that they produce the functions based on the intersections between brain structure and brain function. So of course I can't advance now, but. No, I'm good. Stop, sit, relax, sit. So I'll go through the applications itself, some examples, and then talk about future work. And the future work actually tries to pull together everything across the lifespan. So, and I mentioned, this is the slide I was going to tell you about the team. And I have on one side are the scientific contributions. This is people spanning across the globe. And on the other side are the people that are responsible for actually making the thing work. So on the top, you have the people who are part of the company in Frankfurt and in Romania. And the rest of the people are people who have contributed in terms of software engineering, as well as communication and usability. The platform, as I mentioned, is in fact open source. So if you have a web browser open now on your laptop or your phone, you could actually download the package as we speak. It is written to work on all major software platforms, so Mac, PC, as well as Linux. You can run it on a laptop, you can run it on a high-performance computing system. So it's quite flexible that way. It also has a graphics user interface using OpenGL. So everything can be done under an OpenGL wrapper. I did mention before that the purpose behind VirtualBrain was to try and knit together data. You can run the model without having any data at all. There is sort of a John Doe brain inside that allows you to run the platform and create some cool pictures or models or simulated data, if you will. The way it works is you've got this workflow here. Over here, you've got geometry. So the first thing about defining a brain model is defining the space within which this brain exists. The second thing is, since parts of the brain are connected to one another, you want to identify what are the connections, what are the roads, what are the wires that link different parts of the brain up. So you can define what this is called an adjacency matrix, which is just a fancy way of telling you what's connected to what. So you can define the wiring based on, again, existing information that's there. Or, as I mentioned before, using a thing called diffusion tensor imaging, which is a method to measure white matter in the brain, identify the potential connections between different parts of the brain. So you've got the geometry, you've got the connections. The next thing you want to do is try and impart some dynamics on there. And the way we do that is what's called a neural mass approximation, which is basically a summary of a population of neurons. So instead of being able to measure each individual neuron in the brain, which would be practically actually infeasible at the moment, at least in a human, you want to approximate what you might measure from a collection of neurons, a population of 100,000 or 1 million neurons. So it's called mean field approximations. It's used generically across a lot of physics, specifically in neuroscience, to be able to approximate things like populations of excitatory and inhibitory neurons, for instance. In the virtual brain, there are about 26 different mean field approximations, everything from very simple oscillatory mean fields to much more complicated biophysical models that have cell populations in there as well. So the choice of the mean field is the choice of the user. What do you want to try and model? Keep in mind, this is a modeling platform, so you have to make some decisions a priori. What is it you're trying to understand? What are you trying to model in this brain? So in addition to the neural mass that these generate the dynamics, the activity that you're going to then propagate through the system. You can stimulate individual parts of it, either focally or with some approximations, things like TMS, transcranial magnetic stimulation, can be approximated as well. Deep brain stimulation can be approximated in this platform. And then using what's called forward models, you take those activity that's generated in the neural masses and convert it through what is called forward solutions to things like EEG, MEG, or functional MRI. And you can use that then as a fitting metric to optimize the parameters that are in the neural mass approximations. So you have parameters that govern how the dynamics evolve. You can use the fitting algorithm to optimize those parameters so you're fitting the data directly. So now you're all quiet. Can I have the dog back? So this is the generic equation for the virtual brain. It is there for all different implementations of the mean field approximation. This idea of having a large scale system was something that was introduced by Victor Yersa, who's one of the co-developers in and around 1996 in his PhD work. The generic equation goes something like this. You've got a change in a population measure. This would be local field potentials, is a function of the current state of the state variables. That would be excitation inhibition parameters, for example. The relationship of that to adjacent regions, so how interactions happening between local populations or local cells. And then the input of long range connections. And that would be a scaling factor. You'd have the long range connections would actually have a distance estimate or a time delay embedded in there. So the adjacent interactions are instantaneous interactions. The long range interactions have a time delay. And you can imagine that would be important because you've got here and here, the time delay between this part of my brain and that part of my brain is different than between these two parts of my brain. And those time delays are quite important in terms of how that function that these areas are carrying out is enabled or compromised, for that matter, in the face of damage or disease. So the approximation allows you to incorporate time delays directly in the model. This representation shows you would be the local parameters, the local models for the cell model that would have excitatory and inhibitory cell populations. So you'd have a state variable for excitatory activity, inhibitory activity, their mutual influences, and then the short range inputs here would be other populations. That cell population, that model cell population would be embedded in a region. And then you'd have the long range connection, which you see here. And that would be estimated in terms of the distance from things like diffusion tensor imaging. So you get an estimation of the distance of the connection that would then be used to calculate the time delay. So it all gets put into this relatively elegant yet simple model. Now, it was originally put together to do models of humans, but it's quite easy to apply it to any other system, really. All you really need is some measure of geometry of that system. So the first advance was to use the platform to look at a virtual mouse. So there we used some data from the Allen Institute across the border and derived their mouse brain atlas and put their digitized mouse brain atlas into TVB, the virtual brain. And from there, you've got the connectivity that's in part of the Allen mouse brain atlas, as well as different scales. You have much more information in the mouse brain than you do in the humans in terms of available information. So you actually can get some information about cell architecture and so on, but that can be pushed through exactly the same pipeline. You have geometry, you have connections, you have dynamics, you can generate activity, and then either map that back to empirical data if you're measuring, for example, fMRI in a mouse or potentially even calcium imaging, and then fit those models directly. Also, my colleague Kelly Shen, who's part of the Institute as well, developed a macaque version of the virtual brain. So this was using some data from some colleagues at the University of Western Ontario to generate a macaque atlas and then generate a simulation of the macaque brain. And we're also working now on a marmoset version that will also have the same platform but be applicable to marmosets. We do not have Aplysia or Hermesenda yet, but we'll get there at some point. For learning, the VirtualBrains site itself does have a lot of educational materials in there. There's also shared or mirrored with what's called INCF, which is a Neuroinformatics Consortium, National Informatics Consortium. INCF, in addition to having all the VirtualBrain tutorials, which includes recordings of the lectures, plus all of the material that was used, which includes toy models you can use to learn the platform. INCF also has a lot of other material for learning things about neuroscience, all the way from very core mathematics around spiking neurons to how to manage data, for example, which is becoming increasingly an important thing in neuroscience research. And finally, big science continues to stay big. So there is a new, actually this is an inherited consortium called eBrains. eBrains is a large collaboration at the European Union. It's an extension of what was called the Human Brain Project, which you see the logo down here. Human Brain Project sunsetted September of the previous year and transitioned now to what's called eBrains. And eBrains is an open platform. It is in the European Union, but you can get an account and access that platform and do more that and much more than what's listed there. So you have access to the virtual brain models, but also access to cellular level models. There's actually some in there, it's not listed here, but actually some robotics applications as well, in terms of the code that's used to drive a robot. So VirtualBrain is now integrated in eBrains, which then allows VirtualBrain to be a part of a much larger neuroscience community. So I'm going to transition now to the applications. I want to leave you with one point, which we might come back to depending on how tired you are, which is what is the modeling approach for VirtualBrain versus other kinds of modeling approaches. And so right now, of course, there's a lot of activity and discussions around things like machine learning and artificial intelligence, generative AI, for example, those kinds of platforms. Machine learning is going to be used extensively to solve all of our problems because we can predict up the yazoo. The question is are we actually learning something about the system we're trying to predict? So there are generally two different ways of approaching. This is extreme. It's not meant to be a dichotomy, but it's worthwhile for illustrating the different approaches. What's called generative models and predictive models. So most of machine learning is in the predictive model domain, where you grab a lot of data, run it through sophisticated algorithms that learn the relationship between these data to predict an outcome. Outcomes here could be diagnoses. Outcomes could be where do you put your money for finance, for stock market investments. So it predicts outcomes, but it doesn't really tell you much about the mechanisms that underlie that prediction. Why is it that this constellation of things, for example, smoking, not running, eating bad, results in heart disease? And that's okay because you're learning to predict something, but you don't really know the mechanisms underlying there. Generative models, what you're trying to do there is test hypotheses about what actually produced that outcome, what actually produced in the particular heart disease, what produced that? What is it about the constellation of smoking, not exercising, and eating too much pizza that results in heart disease? What is it about the change, for example, in excitatory inhibitory balance in the brain that might result in having epilepsy? And that approach allows you to build models that generate data. And then the question is, does that generated data produce the phenomenology you're interested in trying to explain? So it tells you about the mechanisms, but it's hard to know how well it actually works. So you really need to assess model fit. Virtual brain is essentially a generative model. So we're taking together and generating hypotheses about certain aspects of brain function and using the model to test those hypotheses in terms of what comes out of the model. So let's walk through some examples. The one that was probably the longest effort was looking at intrinsic activity in the brain. How many people here are familiar with functional MRI as a technique? You can put your hands up, don't be shy. Okay, thank you. There's about 3. No, I'm kidding. So functional MRI has been around for, as I said, about 15, 20 years. It's been a workhorse for studying human cognition. And one of the spin-offs, if you will, of functional MRI is the idea that you could just have somebody slide into the scanner, have them lay down on the scanner, think about nothing, and measure activity and get some information about their brain integrity from having their in a resting state. So if you Google resting state and fMRI, you'll get about a billion papers at this point in time. It's kind of, from a historical perspective, it's kind of funny because it's almost as if they rediscovered that brain has spontaneous activity, but we'll give them that. The question becomes, what is it about that pattern? What's causing that pattern? What are the mechanisms that generate this intrinsic activity? Now, the thing about the intrinsic activity in fMRI at a whole brain level is there are patterns. It's not as if it's random. There is some systematicity to it. So the question becomes, what is that systematicity coming from? Come on in. So the first model hypothesized that what we're looking at when you're talking about intrinsic activity and some systematic patterns is it's an intersection of inherent noise. There's a biophysical noise that's in the brain and some reweighting of connections. So I mentioned earlier, for example, when you incorporate connections in the brain, connections have a distance, time delay, which means that the patterns that emerge can't be all the same thing because the time delays are not the same across all the brains. So you're going to get variation in the spatial patterns. So the hypothesis, what you're seeing in intrinsic activity reflects both that noise and the geometry. And that noise and the geometry reflects exploring configurations in the brain. And that was what you see on the one side of the slide, that exploring the dynamic repertoire is in quotes. What that reflects is an optimal connection strength and connectivity plus noise, which is a system in what's called a metastable state. A metastable state means that you have a number of possible configurations that you can explore simultaneously. Meta in the sense of having a lot, many different states, many stable states that are available. And that metastability results from the interaction between anatomy and function. And that metastability is what you see in spontaneous activity. And what we found was that if you start scaling things like noise, you go from having sort of random activity to having the emergence of these coherent networks. And part of the system that pushes it through these coherent networks are what's called the default network. There's areas that have a special property in terms of their geometry. They have high degree, they are called hubs in the brain. And these areas seem to be the ones that are capable of pushing or pulling, if you will, the networks through different configurations. So the idea is that by having the noise and the geometry, you can explore more of the possible configurations. To make it much simpler, you can have a very simple 3-node system. And when you look at their phase plot of this activity here, you have a system with no noise, and you ping the system, they will oscillate and then come to some sort of quiescent state. You put that in phase space, where I'm just plotting these 3 lines in an X, Y, and Z plot. So you see this nice sort of manifold. If I ping it, it will traverse that manifold and then come down to what's called fixed point. If you add noise to that very simple system and do the same thing, what you find is that the space that network covers as it converges to that fixed point is much broader. So it explores potential configurations as you have that noise on top of some connection profile. So that was the explanation of what this intrinsic activity that has some systematic pattern might reflect. So the question then becomes, does it have some purpose beyond that? Does it have any sort of advantages? And this is I keep on meaning to change this slide, I never do, but So I'll just remind myself yet again that I should make a better slide from this one. But I will at least have, try and walk you through it a bit more sensible way. This is actually a review article that was published in Trends in Neuroscience a long time ago. And it's showing a couple of things about scaling these different parameters in the brain to produce activities that maps back onto what you see empirically. So what you can do here is, as I scale parameters in this model, I can generate the simulated data. I can generate fMRI data for that matter, and compare that directly and get a fit index. And this fit index is basically just a simple correlation between the simulated fMRI data and the empirical fMRI data. So if I do this here, what I'm doing across the bottom part of this axis here is now I'm just changing how strong the connections are. As I do that, you see that the correlation between the empirical and the simulated data starts rising, keeps on rising as I keep on increasing the coupling, keep on increasing the connection strength, and then it drops. You can think about what that might mean intuitively, because if I have a system that's loosely connected initially and I put in this random noise, there won't be much correlation because everything's doing their own thing. If they start connecting them, they're going to start forming networks which will start imparting these correlations. But if I make it too connected, you end up having a single system because everything is so tightly connected that it's just kind of oscillating together. And that's not what's happening in the brain. So therefore, you get that drop in correlations as they get too coupled. If I look at the behavior of the system in what's called phase space, and this is what's on the bottom of the diagram here, you can just skip this part in the middle for now. You see that prior to this first part, I have basically a single attractor. It's kind of like going back to that previous diagram where I had the system with very little noise and it just kind of spirals nicely and then comes to a single fixed point. As I start changing the connections that have more networks emerge, you have more attractors emerging. You have multi-stability emerging. And that's what you see in, in between these two areas here. This is the emergence of different attractors, different parts of different potential configurations. And then when I go too far, it all collapses and you go back to a different kind of attractor space. So this is kind of like the sweet spot in here. There's interesting stuff going on here. The question is, what does that mean? Like, what happens in that thing functionally? So if I go ahead and ping that system in that sweet spot, so for example, I put a current or a stimulation, for example, in visual cortex and see how that signal propagates. What you find is that in the first part of that system, when I first cross that barrier and I put a signal in there, it propagates at some speed and then tapers off. But move a bit further, it actually propagates much more rapidly. So that means that being in that sweet spot, the metastable state, allows signals to propagate maximally through the system. So that's the benefit of having both noise, this optimal connectivity, and having this metastable state. A lot of stuff I just said, I know, but that's essentially what came out of that series of studies. That this intrinsic activity represents the balance of noise and connectivity that leads to a metastable state. Metastable, again, being the availability of many different kinds of networks that the system can utilize then when it's faced with a challenge. When it's faced with a challenge, that challenge or that ping propagates through the system at an optimal rate. And that's what the benefits of having this confluence of noise and anatomy. And that's the benefit of having the generative model, because it does in fact give us some information about how those variables interact. I think what I'm going to do, this is just more technical stuff that makes it very complicated, but I will mention that it's nice to see that these ideas do come up in the literature. So now there's, again, large projects that are looking at big data sets. This is the Human Connectome project, and this is about 1,000 individuals that have been scanned, very high resolution scanning, looking at resting state fMRI, but also different imaging technology, imaging methods, DTI, for example. And what you find is that here as well, when you look at resting state activity and put it in this manifold space, you find that humans, not simulated humans, will in fact show the same exploration state. And you can then use those data to create individualized models to fit what you see empirically to those data and show that there is in fact variations across individuals as how well they explore this space. And there are consequences in terms of the cognitive functions that they're able to support in terms of how they traverse this resting state space. So it is providing both a foundation in the terms of explanation of what's happening there, but also then be able to use that then going back to empirical work to help understand why you're predicting changes in cognition or not. So the good part about our intrinsic evaluation thing was that there's very clear motivations for the models. It does help to develop theories, and it can be the driver, as I was mentioning, for developing new applications, which I'll mention in a second. The bad thing is, as I mentioned at the beginning, there's 26 at least different kinds of mean field approximations. We did not compare all 26 models to see how well they worked. So the choice of model really depends on an individual, which you think is the best way to approximate different kinds of features. Across those models, the use of time delays and nonlinearities is not systematic, so that could cause some issues going forward. And finally, the dependencies on data fitting is not clear what features are used for data fitting. But that's also a project that's going to be someone's PhD thesis at some point in time. Let's talk about stroke recovery. So this is the first clinical application. What you see here is a brain from a person who's had a stroke. You can see the stroke here on what's called a T1 image. So there's a lesion in the left hemisphere. And a T2 image shows you also the lesion here. The goal here was to see whether we could, first of all, actually model data from stroke individuals, but then also can we use the parameters from the model, measures of excitation and inhibition, for example, in cell populations, as ways to predict how well someone was going to recover from that stroke or respond to therapy. So the model that was used was the same kind of model I talked about earlier: population activity, short range input, long range input. For the application here, we focused on the balance of excitation and inhibition plus the long range interactions. What we did was took the individual subjects' functional MRI data and started doing data fitting and looking at the parameter space and where do you get the maximum fit to the model. So for example, here is a healthy subject. And what we're scaling in this particular case, go ahead, is global coupling and conduction velocity. And you see some hotspots coming up here, and that's where the fit is maximum in the healthy control. On top, we have a few more examples from healthy controls, and here we have some examples from 3 different stroke individuals. Some stroke patients show very similar parameter spaces to healthy controls. Others do not, suggesting there's a change at least at the large-scale network level. Not surprising, since they do have a big lesion in their brain. But then the question becomes, can we use those parameters further? And the answer is yes, but I'll show you that in a second. This is the local parameters. We have excitation and inhibition. And again, parameter sweep. So we're changing parameters, and you find here's the sweet spot. This again is a healthy control. We do it again across the different sample. We've got healthy controls here. We've got stroke individuals. Some look like controls, some do not. And now we look at how those parameters relate to clinical metrics. So here we're looking at the fugal maintenance and post-therapy measures in motor rehab, and the correlation between their scores on these tests and excitatory coupling, and find that even in the small sample size, you find that the parameters those individuals have in the local cell parameters, excitation in particular, predicts how well they're going to respond to to therapy, suggesting there's actually some diagnostic or prognostic utility in doing this. This is a small sample, so we have to also just follow it up, but at least suggest that there is some potential to use this to help guide how you might intervene. So for stroke, again, good example of prognostic utility. There is a direct connection between the parameters that were used to do that prediction and what you see in actually physiological models of stroke. So you do see both in animal models, but also in clinical studies, that a common consequence of having a stroke is hyperexcitability, at least epilepsy, for example. So you do see seizure activity in and around the stroke itself, and that comes from hyperexcitability of the remaining tissues. And that seems to be bad in terms of outcomes later for therapy, for instance. We had a small sample, so that's not great. We actually have to show prognostic capacity beyond the initial follow-up. And again, getting back to does that change if we change the mean field approximations? Don't know. Let's talk briefly about dementia. How are we doing for time? I know our time's all messed up. I don't want it 10 after 3. But what I'll do, so I give Sabrina some time for her class. What? Can you? Okay. Then I'll keep going. Keep on going. So dementia, of course, is a big issue. And dementia research in terms of big data has seen an explosion in the application over the last decade as well. There's a project called ADNI, the Alzheimer's Disease Neuroimaging Initiative. That's actually a global collaboration that's involved in acquiring data from mainly Alzheimer's patients at the different levels or different severity of the diseases. All the way, it started in the US, but it's actually now involved China, Japan, Australia, Europe, and some sites in Canada as well. There's also the complement is the Sydney study that I'm mentioning here. That's also a prognostic study. And the first application to dementia was to use a subset of those data to look at controls, to look at persons who had mild forms of Alzheimer's, and then persons who had mild cognitive impairment. Mild cognitive impairment being the state at which you don't really know if they're going to be Alzheimer's or if they have another kind of memory disorder. It could be they've had a stroke that's not, you can't see on the scan. It could be they have other kinds of dementia, Lewy body, Parkinson's dementia, other sorts of memory impairment. And so the important thing, of course, is when someone has Alzheimer's disease, it's pretty clear, relatively speaking. If you have MCI, you don't really know. So the question there becomes, can we use the model then to help stratify people who have MCI early on? So the workflow is not different from what I've talked about earlier, but just to remind you that we're getting measures of connectivity, diffusion tensor imaging. We're getting resting state fMRI from these individuals as well. We're putting it into the VirtualBrain platform where you put in the connections. You have the mean field approximation looking at excitation and inhibition. So each node has these little cell populations. You have the simulated resting state fMR, simulated functional connectivity, which is just correlations. And then you do data fitting between the simulated functional connectivity and the empirical functional connectivity. And then look at the parameters from that model with respect to, in this particular case, cognitive functioning. I'm just going to skip to the next slide. So if we focus on sort of this last part here, this tells you the relationship with the parameters in the model, so global coupling, inhibition, excitation, excitation, inhibition, and these self-regulatory for excitation itself. The bars are just basically confidence intervals telling you those correlations are stable. These are the different cognitive measures here. And what you see down here is the spread of individuals in terms of healthy in black, Alzheimer's disease in red, and in the middle you have the MCI individuals. And this is really what the important part here is are all the MCI individuals, are they scattered more on one side than the other? And can we then learn something about that individual patient in terms of are they showing Alzheimer's type dementia? Are they showing other kinds of dementia? And does that help us then, again, stratify them in terms of where they're going? And you do, in fact, see that they are scattered throughout here. And since this is a prognostic or a prospective study, I should say, we can then go back and track those individuals who had MCI and see where do they end up as we're going forward. So it does suggest at least that it's able to stratify them. This is just an example showing you that if you do use the model with VirtualBrain as the fitting feature, that you do a much better job of accounting for variance or covariance with the cognition than with other measures like functional connectivity and structural connectivity. Telling you that the model, using the VirtualBrain as the model, actually does much better in terms of predicting. So again, good proof of principle. We can extend that even more now. There are other imaging modalities that are used in dementia quite frequently. One of them is amyloid imaging to look for deposition of tau and amyloid beta. This is done using PET, positron emission tomography. So what you could do is use the PET image from the individuals to look at the depositions of these toxic proteins. And that may tell you a bit more about the distribution of network changes in the brain and potentially then use that to help guide interventions. So here again is another proof of principle is that we have amyloid beta PET. We can then change the mean field approximations for regions that have high amyloid deposition to change their excitability. So what amyloid beta does is it changes the excitability of cell populations. So it downregulates inhibitory neurons and makes the cells basically hyperexcitable. So you change the excitability ratios in those regions. One of the early treatments for dementia that's used when a person shows probable dementia is given them memantine. Memantine is a drug that basically counteracts the excitability and helps to reestablish the balance. So we can do that virtually here as well. We can give the VirtualBrain memantine, generally speaking, and see, does that then reproduce what you see in terms of changes in activity that follow memantine to a patient who has dementia. If we look at the spectral power curves, this is basically a forward solution, again, generating electrical activity in the brain. You see what the electrical activity is in healthy controls in black, in MCI in green. In Alzheimer's patients without the memantine that have this deposition, you see that the spectral power is actually reduced in certain frequencies. When you give them virtual memantine, give them virtual therapy, you see that their spectral power recovers, at least initially, suggesting that you can, in fact, model drug interventions now. So now we have a way of doing potentially prognosis for dementia, and then being able to model what the effect of giving a particular drug could be for an individual in this as well. Again, it's a pretty good collection of pieces. So for dementia, again, we have good examples of prognostic utility. We do see that the parameter ranges you see in AI balance changes in dementia do map on quite nicely. And we show a way to integrate even more information, pulling in some more information about the protein deposition as well as pharmacological interventions. The bad thing is obviously we need more longitudinal data to be able to track people who go from MCI to dementia. The second point about not modeling neuronal loss, it depends on who you ask about that. It's not easy to model neuronal loss when you've got a population that's a single number, but there are ways of doing that. And the last thing is that for those who are going to go into clinical research, if you see a person who has dementia, it's seldomly a single thing that causes that dementia. There are very few pure cases of things like Alzheimer's out there. And so even though the ADNI data set that I mentioned is putatively pure Alzheimer's, most of the time you see people who have multiple etiologies. And more often than not, it's usually vascular contributions on top of that. And that just complicates the game. So it has to be incorporated somehow. The most successful application to the VirtualBrain thus far has been to epilepsy. I'll tell you why. Because I'm sure you're asking that question right now. Why is that, Randy? Well, I'll tell you. Epilepsy is extremely well researched, not well researched, but heavily researched in dynamical systems theory because it has these very interesting cascades of activity that can be described phenomenologically, as well as through other more biophysically based models. The person driving this project is my colleague Victor Yersa in Marseille. He is a theoretical physicist by training, very much in dynamical systems theory. And when he was shown this trace down here, which is a trace from a rat hippocampal slice, and this is showing you the spiking pattern, but also some slow currents down here. What he saw phenomenologically was that this represents two different processes, of course. Of course, they say. What it first represents is you've got a fast process, which is spiking. So you've got these spikes that are happening here. And then as the spikes dwindle, you've got a change in some sort of enabling variable, or what's called a slow variable. So you've got a slow variable acting here that changes its values, and it changes its values, it actually sort of squashes, if you will, spiking activity. As the slow variable oscillates back up again, then enables the spiking to re-emerge. Goes back and forth, so we have the spiking activity, which is the seizure, and then resting state, spiking, resting state. Put that in the phase space again, you've got the slow variable changing. You have what's called an entry bifurcation. You have what's called a spiral attractor now, generating spiking activity, exit bifurcation, resting state, circles back. Very nice, very simple, what's called a phenomenological model. It doesn't have a direct connection to a biophysical parameter, like for example, calcium concentrations, but it does produce quite nicely the phenomenology that you might want to model. So what we can do is take, and instead of putting in a mean field approximation that has excitation inhibition, we can put in a mean field approximation that has excitability, how easy it is to transition into that spiking behavior into the model. And so what Victor and his group did was take the same geometrical representation for a patient, where you get their T1 image, you get their diffusion tensor imaging, you also get some measures of intracranial EEG that's used to diagnose the likely location of the seizure, and use that as the fitting feature. And then walk through that collection of different nodes and modify excitability to say where is the likely location of the seizure focus. Because the game in epilepsy, not really a game, but the target in epilepsy surgery is to identify where is the seizure focus and avoid the areas that are propagating the seizure. So you have the epileptogenic zone and the propagation zone. You want to avoid propagation zone and get the epileptogenic zone, because that's really where you're going to get probably the best outcomes if you can identify it cleanly. And so again, just to walk through the diagram here, you've got your diffusion tensor imaging. You've got your CT, which tells you where the electrodes are. You've got your intracranial, what's called SEEG or stereotaxic EEG recording. Put it in the virtual brain, generate activity, and do what's called model inversion to identify where is the likelihood of these different seizure foci. It then gives you a map of where the epileptogenic zone is, and you can then use that potentially to guide interventions, and then you can actually do that directly, and I'll tell you about that in a second. This is a movie of a virtual brain from a patient. So on the top, you see the clinical information that goes in there. The bottom is simulated data. And up there, you see the balls that are getting larger and smaller. Those are simulated electrode locations that correspond to these electrode locations and telling you how the seizure is propagating. So this is the onset of the seizure here, moving along that way, and then it propagates to the other hemisphere. Let's see if that circles back or not. No. I'm not sure if I can start again. Yeah. So again, seizure starts, you can see the balls lighting up, so it's propagating a bit more. You'll see it spreading. And the cool thing about this is you see where the yellow circles correspond to the electrodes that are actually implanted in the patient. The blue dots correspond to sites that are part of the model. They're actually nodes, but they're not really measured, at least clinically. And what they have found in applying this is you do get situations where what the model suggests is the epileptogenic zone is an area you did not measure, which is important information to know because you might say that you need to either go back in and redo the measurement or find a different way to treat this individual. And so what they found in some initial evaluations was, in fact, where you do have good outcomes in a retrospective study is where the seizure focus that was extracted in surgery was, in fact, very close to where the virtual brain suggested it should be. And the poor outcomes was actually where there was a divergence of what the clinicians decided to extract and the regions that were actually suggested to be seizure foci in the individuals. And that was, in fact, in this case, down at the bottom was one where the surgical outcome was not great because they extracted areas here, and the seizure focus was actually more ventral as well. That, as I said, led to a successful outcome for virtual brain and epilepsy because it led to a national clinical trial that's just finishing up right now in France. It's been going on for 5 years. It's a prospective study where they're using virtual brain as a decision support tool for surgical intervention. So they're now using that. They give the clinician both the regular clinical follow-up, but also the probabilistic locations for different seizure foci. And that becomes some of the discussions now that happen with the neurosurgeons in terms of where do I now intervene in that individual to prevent the seizures from happening again. This can be used for surgery. It can also be used for stimulation, which is a new technology is being considered for epilepsy. Instead of having excised tissue, you can actually stimulate locally and try and avoid the seizures from happening altogether. It's a little bit less invasive than having to cut open the head and then taking things out. So again, the epilepsy evaluation is an excellent example of translation from fundamental and theoretical neuroscience to clinical decision support. And it has improved not only the generative model aspect, but I didn't mention is this, the reason for the success of this is a confluence now between the generative models I mentioned earlier and a predictive modeling approach. This particular case is actually what's called Bayesian inference to identify where is a likely seizure location using Bayesian estimation. And even though it's phenomenological, it can be linked to biological processes to drive further fundamental research in terms of how to understand these slow and fast processes in the brain. The challenge, of course, is that this is a great example of successful clinical translation. It's not really clear whether the same pathway is available for other clinical applications. There's a paper that's coming out soon in National Science Review that outlines some possibilities, but there's lots of work in progress to be done. Again, many more PhD theses to be discovered in that domain. There's a question about workflow scalability. As you can imagine, this is quite labor intense. The aspiration, of course, is to make this compact so it can be part of a standard clinical practice. And that compactness is something that needs to be worked on a bit. And there are some issues with model inversion as well, but that seems to be a solved problem because of improvements in the optimization routines in computer science. I think what I'm going to do, so the work's in progress. I'm going to skip some slides here, but I want to just mention a couple of things which tie into thinking about the prediction, the intersection of prediction and generative models, and how one might use this then to really expand the collaborations, if you will, between computational modeling empirical neuroscience and ultimately clinical decisions could be done. So think about the epilepsy example where we generated a whole bunch of data now from people who've had epilepsy surgery and looked at their outcomes. You can imagine that getting scaled now. In that particular clinical trial, they had about 400 patients, but you can imagine if you did that across all of Europe or across all of North America or across the entire planet, you'd be generating a population of patients. You'd have good indications of their outcomes by knowing what the models did. There's no lack of large data sets that are out there for other things. For example, just healthy development. So for example, in the Human Connectome project that I mentioned earlier, there are now initiatives that tag on both the front and back end of adulthood. So now you have the developmental part of that. It goes from kids from about 5 to 15. And you've got now the older adults from about 40 to about 80. And that Human Connectome project has a longitudinal component. So what you can envision happening now is as we collect these data, they themselves can be ways to generate models of individuals that have biophysical parameters that we can modify and say, if I have somebody who's 20, how do I make them look like they're 40 or 50? And can I make them go backwards, for example? Can I do anti-aging? You could also then link that to their cognitive profiles, because a lot of times you have very similar cognitive metrics that could be used then to define a parameter space. So a parameter space could be something like this, where you've got a collection of virtual brain parameters that could be, again, our excitation inhibition balance, our network connectivity, either the conduction velocity or something thereof, and relate that to cognitive function. You can then look at different parts of that parameter space and say, what does the physiology look like there? Does it look like it's actually garbage? Does it look like something that's actually reasonable, but you don't see that very often? If I don't see that very often, what does it mean in terms of cognitive function? Could it be, for example, that this is a pathway to dementia or cognitive impairment? Or is this actually a pathway that suggests that that's actually more resilience? We're looking at the cognitive status. So it's a way to understand the span of cognitive diversity. The span of cognitive diversity underlies sort of healthy brain function, but also the cognitive diversity might lead to other bad clinical indications. You could then envision expanding that to things like dementia, where you've got your healthy control model, and then you have some sort of insult, whether that be stroke or it could be a dementia, where you actually have a transition, like too much deposition of A beta or tau, or too many Lewy body systems, or stroke. And then you have a bifurcation that has these different trajectories. And then use that information in the field, because you could then generate simulated data that would be an indication of somebody who has dementia and have that as a fingerprint. It's almost like if you go to a physician and have them listen to your heart. Physicians typically learn what a good heartbeat sounds like versus a bad heartbeat. They don't know why it's bad. They just know that it is bad. So they have sort of a low-res way of measuring your heart. But you can think of having the same kind of low-res way of measuring your brain using clinical EEG, but knowing that these patterns reflect either good or bad trajectories, and use that again as a way of doing early diagnosis, early stratification, and early intervention. So I'm going to leave you with the general overall good and bad stuff about the virtual brain. The virtual brain is model agnostic in the sense you have all these different mediable approximations, some 26 that you can use. It does support inferences on mechanisms, so you can make inferences about connection strength, excitation inhibition. You can work directly with empirical data, which helps with clinical translation. It's open source, open access. It's a community project. We do have the industrial rigor that provides good software support. And I think still that we have probably the best logo out there. Because like this is cool. And when someone sees that, it's like, you can see it, put it in my head almost right there. See? It's probably one of the most recognizable logos that are out there right now. So I like that. The bad parts is the model fitting and identifiability. That's not unique to virtual brain. That's true for almost any kind of dynamical systems modeling. But it is something that we have to pay attention to. There are ways to get around that. Neuromodulation, that's an emerging aspect, looking at things like dopamine, norepinephrine, acetylcholine. All the models right now are brains in a jar, so we don't have them actually doing anything. So obviously putting in function, whether that be cognitive function, motor function, or actually thinking about brains that are interacting, that are listening to Randy blabber on about virtual brain for the last hour, for instance. That's not part of the model yet. There are some questions about translation between the population level of the physiological measurements and getting down to single units. And that's something that's, it's possible, but it's not easy to do. And finally, interpretation is always a challenge. And that's not necessarily, again, unique to virtual brain, but it does add a different level of thinking about your data because you're going from what you've measured, fMRI, to both that plus the underlying physiology, connection strength, EI balance, things of that sort. So good and bad, but overall, I think overall it's actually pretty good. So thank you. And you will get course credit for this, Mr. Breen. So thank you all for your attention. Appreciate it.